Okay, thank you, Himangsu, for the introduction. Hello everyone. I'm going to present my recent work about the recent publication about the diazotrophy in the Indian Ocean, about their current understanding, what is happening in the Indian Ocean, and with the future perspective. So before going to start I would like to let you know that our project DINDE and IDAFIX, that is endorsed by the IIV2 Second International Indian Ocean Expedition, and also I would like to thank IIV ECSN for giving me this opportunity to present my work. So I work on nitrogen fixation. So as a nitrogen fixation is one of the main exogenous source of nitrogen. And what it happened actually, this convert mainly the inert dinitrogen gas to bioavailable ammonia. It's an energy intensive process, and that can sustain the productivity in the oligotrophic Indian Ocean up to 50% to the new production, but it varies at different basins. It also helps in the carbon dioxide sequestration. Like if you look here into the dinitrogen gas with the help of nitrogen fixation is coming into the mainly ocean ecosystem as in nitrogen, and this helps in fueling the productivity, and also these are incorporated into the ecosystem. So they act as kind of a fertilizer in the ocean. And when this system is breakdown here, all the system will be collapsed because as it's continuously connected to the higher trophic level. So who fix actually the nitrogen? So this organism is called diazotrophs. It comes from the Greek etymological term, which is di is two, azo is nitrogen, and trophos is the nutrition. And they fix the nitrogen with the help of the enzyme nitrogenase. It has mainly two components. One is the nitrogenase reductase, that is the electron transfer side, and it's encoded by the gene NIF-H, whereas in the other side there is dinitrogenase, that is the main catalytic site where fixation happens, that is encoded by NIF-D and NIF-K. If we look into the diversity of diazotrophs, now earlier it was thought that cyanobacteria is the main diazotroph that fix nitrogen, but with the growing evidence and with the recent advancement of the molecular tools, we came to know that it's much more diverse than what earlier people thought. Like cyanobacteria just represented a plate, a portion of the whole phylogeny, where there you can see there are a lot of other different groups. These are collectively termed as non-cyanobacterial diazotroph or NCDs, and I'm going to talk, like in the following slides, I'll talk about like also the NCDs. What are the different? And on the right side, you can see, and these NCDs are mainly the alpha, beta, gamma. These are mostly heterotrophic groups. Heterotrophic, we call them heterotrophic diazotrophs. So if you look onto the right side, there you can see it's a complex phylogenetic network that represent only the diversity of NCDs. So even within NCDs, there are a lot of different groups and there are a lot of complexities in their genetic makeup. Overall, the global picture to look into the where our Indian Ocean is standing, like the first global compilation from 2011 and 2012, where the Indian Ocean was not included because there was less number of data points as compared to the look, and mostly the data are compiled from the Atlantic and Pacific side. So Indian Ocean was totally discarded. Again, there is in 2012 where they extrapolated the rate from the North and South Pacific to the North and South of the Indian Ocean. And with the model, they predicted Indian Ocean contribute 26 teragram of nitrogen to the overall nitrogen budget. And there are subsequently two papers from one is model-based prediction and another global compilation by Tang and Kaiser, where with the model-based prediction, they said that Indian Ocean is contributing 11.5 teragram of nitrogen per year into the global budget. Recently, there is a compilation and it's published like last month, and where you can see they tried to incorporate more data that was earlier not added into the database to overall to know about how Indian Ocean is contributing to the global budget. And their compilation suggested that Indian Ocean predicted 35 teragram of nitrogen by arithmetic mean and 7.1 by geometric mean. So to know about the factor, what are the different factors that affects the nitrogen? On the right side, nitrogen fixation. So on the right side, this is the image of a nitrogenase enzyme. There you can see there is two main component. One is the iron protein, that is the NPH, the dinitrogenous reductase, and another one that is the MOFE protein, that is the catalytic site, where there is a iron sulfur. Also in all these cases, there is an iron sulfur cluster that is binding these two portion of the protein and helping in the nitrogen fixation. So this nitrogen fixation is one of the there are different control mechanisms, that there are biogeochemical control, there are physical controls. So one of the major, the basic control mechanism comes from the oxygen, because oxygen inhibits this enzyme. As you know, there is the energy demanding process. It requires 16 ATP. So energy could be also a limiting factor. Other than temperature, like this nitrogenous enzyme effectively work in an average like about 25 degree or near 25 degrees centigrade temperatures to be more effective. And N by P ratios, or the NP nutritional control, that's to help to identify the nitrogen limitation into the ecosystem, because these are the place that mostly nitrogen fixation happens when mainly the oligotrophic. Though there are recent evidence in the coastal, where is the high nutrient areas, and from the polar ocean also. Iron bioavailability, as you can see from the enzyme structure, iron is one of the important trace metals along with the molybdenum here. So iron could also inhibit nitrogen fixation and dissolve organic matter. That also helps for the mainly heterotrophic diazotroph to gain the energy to fix the nitrogen. And there could be a molybdenum and sulfate antagonism. If there are more sulfate, that could bind instead of molybdenum and stop the nitrogen fixation, the activity of the enzyme. So why Indian Ocean? What is happening at the Indian Ocean? Recent global climate model that suggested that with the change of climate, primary productivity is going to increase. And this is because of the nitrogen fixation, though there are uncertainties, and uncertainties mainly because of the Indian Ocean, because there are less coverage in the model from the Indian Ocean. And on the right side, this is the 4 different images that is on the background. There is a nitrate biphosphate ratios, and these are the different points like overall area where nitrogen fixation have been measured at different monsoons that represented in the 4 figures. And in the Indian Ocean, there is a strong seasonal influence as compared to the Atlantic and Pacific. There, you can see from the image, there absence of several basin scale nitrogen fixation activity, and from the model it suggested that it could be hotspot, this area for the nitrogen fixation activity. And as there are strong seasonal influence, subtropical to subpolar gradient, so this could be also a potential for different unknown diazotrophs. So to understand what is the problem, to understand properly the nitrogen fixation or diazotrophy in the Indian Ocean. So this is a loophole where you can see the red point is absent. So most of the places in most of the subbasins, so the Arabian Sea, Bay of Bengal, Equatorial, Eastern Indian Ocean, subtropical gyre, and Southern Indian Ocean, there are mostly research focused on the northern areas, and even for only for the biogeochemical stuff, not on the molecular perspective, to look who are the different groups, how they are infecting, or how the biogeochemical condition is impacting those organisms. So these are the mainly burden to understand, because of the less number of coverage, to understand properly the diazotrophy in the Indian Ocean. So what we did, we compiled the 30 years of data. There are more data that is even in the global compilation that is published. There are some missing data point that I can see from also the global data set. So what we did, we compiled the 30 years of nitrogen fixation data that till now available, though there is a recent paper from Himanshu that is not added here. So our compilation is suggested that Indian Ocean is contributing to the global budget 7 to 13 teragram. And this could be more implicated when we will have more coverage from here. You can see this whole portion of the Arabian Sea on the there is a missing data point. There are also a lot of missing places that nitrogen fixation could be explored, and this will help us to understand, to estimate the global compilations, also the Indian Ocean contributions. Overall, the communities, what I did also, I reanalyzed all the data that is available from the Indian Ocean to look into the community composition, how the community is making up, where there are more unique different group that is coming up. So, and there are 3 basins is excluded because there is no data about the about the mainly community compositions. As I mainly highlighted Bay of Bengal, Equatorial Indian Ocean, and Eastern Indian Ocean. And out of that, Eastern Indian Ocean is host most of the unique taxa that would compare to the Bay of Bengal and Equatorial Indian Ocean. This is the different groups that are the mainly unique group that is present in the Eastern Indian Ocean sector. Overall, there is a recent paper publication that talked about the global compilations of the diazotrophy group from the surface of the ocean, and where they found that alpha proteobacteria are more abundant as compared to the gamma, but Indian Ocean is more unique on that perspective. So this is the genetic makeup or like the overall diazotroph community composition. You can see there are more gamma proteobacterial complex and diverse group of gamma proteobacterial diazotroph than the alpha proteobacteria. Overall, the diazotroph community, you can see from here a different basin, they have different type of community compositions. And Bay of Bengal, out of these 3 regions, host most cyanobacterial diazotroph into the community. If we look into the normal like a Pearson correlation factor, and there you can see the nitrogen fixation is mainly driven by the cyanobacterial group, not the non-cyanobacterial diazotroph. So there is no direct evidence of the non-cyanobacterial diazotroph is fixing nitrogen in the Indian Ocean. So this is overall the heat map from different basins compiling different monsoon season to show the effect, how monsoon is strongly affect the community makeup in the Indian Ocean. Like the first one, you can see the intermonsoon, there are more cyanobacterial group, whereas here in the winter, as in the equatorial Indian Ocean, also in intermonsoon, there are more like alpha proteobacteria. This is the gamma proteobacteria along with the, but in the winter, there are in equatorial Indian Ocean, there are more cyanobacterial group. Interestingly, from early inside the even one monsoon, if you look from early to late intermonsoon, like here you can see from the Bay of Bengal, like there is, or in the equatorial Indian Ocean, there is alpha is mainly dominating the community. But in the late intermonsoon, there you can see the gamma. So it's suggesting that even within a single monsoon, there are community shift. So this strongly highlighted the importance of the monsoon impact into the genetic makeup of the community in the Indian Ocean. Also, this is represented at different depths. So there are more heterotrophic group that if you go down to the from the surface, and there are mostly the cyanobacteria are present in the surface water, as then it's also the light. And in the eastern side of the Indian Ocean, in austral summer, there are more cyanobacteria, whereas there are more archaea and gamma proteobacteria during the intermonsoon season. So to give a little bit more context about the loopfall in the Indian Ocean during my PhD, I tried to fill some of the gaps. So here in this, this is the from the Geotraces cruise in 2021 during austral summer in the southern Indian Ocean that covers the oligotrophic gyre to the HNLC region of the southern ocean. So here what we found that interestingly below the frontal zones, like after 42, there are mainly the fronts positions started like subtropical front, Augulus front, Antarctic front, polar front. So below the front in the colder water, there is no nitrogen fixation activity. That is also reflected in the community composition. Here, the black line I put because to indicate the left side is below the fronts and the above are mostly above the fronts like in the gyre or in the Augulus region. So it's reflecting that Augulus is kind of also a hotspot for in this area for the nitrogen fixation activity. There you can see there are more cyanobacterial group by the amplicon sequencing approaches, and also we targeted 6 different groups, mainly 4 cyanobacterial groups, Trichodesmium, unicellular cyanobacteria B, A1, A2, B, and gamma A and gamma 4. So where we also found concomitant high abundance of Trichodesmium and UCY in A1. Interestingly, UCY in A1 earlier not detected from the Indian Ocean. Recently, there is a paper from the eastern side of the Indian Ocean, like Eastern Indian Ocean, they found, but in this part, there are more high abundance of the UC wine A1, and these are one of the main driver of the nitrogen fixation, particularly in this area. Whereas the other group are more like homogeneously distributed, but there is a reflection on mainly the gamma-4, where there is a high chlorophyll consumption, like a chlorophyll. There are some chlorophyll patches, and this part, the gamma-4 is popping up. So to look into the drivers, who is driving the community in this area, we try to look into the compile the trace metals data, inorganic nutrient and physical parameters. And what we found here, mainly the temperature, manganese are mainly driving the cyanobacterial community, and these are helping the nitrogen fixation activity. Even also, we look into the by amplicons and to look why we detected or what are the different parameters that is influencing the different diazotroph group that we detected. Well, there also we found a strong influence of manganese and temperature into the nitrogen fixation activity, and mainly the cyanobacterial group that we detected above the fronts, like in the north of the fronts in the Jaya region. Whereas in the south, there are more inorganic nutrients because it's the HNLC area, and also there are less influence of the trace metals, mainly the zinc, nickel, cobalt. They are mainly influencing the heterotrophic diazotroph group in the southern part of the Indian Ocean. But another word to look into the, because also you have seen there are screen sharing stop. Oh no, sorry. Is it visible now?
Yes.
Okay, okay. Sorry for the interruptions. So I also try to look some of the fill the gap in the northern Indian Ocean, mainly in the Arabian Sea and the Bay of Bengal, where also we found that the diazotroph community makeup is different in the subbasins of the in northern part of the Indian Ocean, where there are more alpha proteobacteria, beta proteobacteria in the Arabian Sea side and Bay of Bengal, and mainly the Sri Lankan coast side. This is the most cyanobacterial group. And there is also, you can see the community is well separated. There is a link of the Sri Lankan coast with the Bay of Bengal. And we tried to look into the abundance, and what we found that the trichodesmium is doing, this is during the intermonsoon, and trichodesmium is more abundant in the Bay of Bengal side, where there are more unicellular cyanobacteria in the Arabian Sea side. So overall, what we need to properly understand. So you can see like there are, I try to fill little gaps, and still there are more gaps to fill, to look into the really in depth and detailed diazotrophy in the Indian Ocean. So for that, what we need more spatial coverage in different monsoon seasons to really look into how monsoon is more affecting the community makeup and nitrogen fixation activity. We need more depth integrated measurements, because most of the depth integrated measurements from the Indian Ocean, they are mostly focused on the northern side. So communities should consider also the dedicated work in the equatorial and subtropical Jaya, also with the southern Indian Ocean sector. Also, we should consider about the trace metals into account, as trace metals is one of the most important part in the enzyme of the enzyme nitrogenase. As you have seen, there are uniqueness among the community of, in the subbasins. So future work focusing on the metabolomics or transcriptomics will help to understand how diazotroph is adapting. I believe that as there are changing, continuously shifting the community at different monsoons, so these diazotroph are much more adapted as compared to the Atlantic and Pacific diazotrophs. So future work with the strain isolation and to look into the different factors inside in the lab will also help to understand who is actually more driving at the different groups that is present in the Indian Ocean. And also with the recent context of the climate change, the Jaya is sinking and extending in different monsoons. So this could also, and will be interesting to look how it was going to impact the diazotrophy. Diazotrophy is extending toward more south or no. So overall, my take-home messages is winter and intermonsoon seasons are for the northern Indian Oceans, and intermonsoon for the Eastern Indian Ocean are likely responsible for the higher nitrogen fixation activity and more cyanobacterial abundance. Total contribution from the Indian Ocean is 7 to 13 teragram of nitrogen per year. And the diazotroph, there is, as you have seen, there is a high spatio-temporal variations into the diazotrophy. And also in the southern side, there is a clear latitudinal break in the cyanobacteria and NCDs in the community makeup, nitrogen fixation activity. And trace metals, mainly manganese, is also indirectly influencing the abundance and the community in the southern Indian Ocean. So overall, I'd like to thank my group, my supervisor, external collaborator of the project, and other collaborators that we work with. And if you are more interested to read about more in-depth, what is the biogeochemical things are changing different monsoon in different sub-basins, you can look into my recent paper. And if you have any further question, you can contact me, or I'll be happy to answer any of the questions. And thank you very much.
Thank you, Subhadeep. Thank you for summarizing the current understanding of Indian Ocean and its diazotrophy. I now invite participants to please ask questions. We have one question here from Danija. She's asking, what are the factors driving the abundance of the diazotrophic community in the Western Indian Ocean? Should I repeat?
Yes, can you repeat, please? Yeah.
What are the factors driving the abundance of the diazotrophic community in the Western Indian Ocean?
So in the Eastern Indian Ocean sector, there are mainly the temperature, as it's very well-known factors. And along with that also, there in particular region, like in the Madagascar, south of Madagascar, there are some iron inputs. I don't know much about the atmospheric depositions, but we have the trace metals data, and trace metals data is suggesting that there are mainly manganese and lead that are influencing mainly the cyanobacterial community and also the nitrogen fixation activity. Iron is not much explaining the diazotrophy in the southwestern part, though in the Indian Ocean.
Okay. Next question is, when you say gamma or alpha proteobacteria, do you include only diazotrophs that fall in the gamma or alpha, or all the organisms?
No, I only include, because whenever I look into the diazotrophy, I look into the specific niphage, not into the whole microbial community makeup. So these are the only group that are the diazotroph inside the gamma and alpha, not the whole gamma and alpha.
Okay. One more question. Are there future cruises or missions aimed at increasing the number of observations in the Indian Ocean?
Yeah, there are future cruises that is coming to cover at different monsoons, mainly for the, as you have seen, like for the northern sector. I don't know exactly in the Arabian Sea, but there are future cruises that try to cover from the southern sector and the western sector of the Indian Ocean. And that is
Okay.
also the part of the people from the IIB too, they are trying to do that. And actually, we need more like the spatio-temporal coverage that I was saying. Like if you look into the, even there are more data on the northern side, on the western side from the middle, even in the OMGs in the Arabian Sea is one of the biggest OMGs in the world, but there is very few data points in the OMGs. And these are the prone areas from the literature that say that these are the most denitrification zone. So this denitrification zone where to counterbalance, there should be more nitrogen fixation. So, but we don't have that much evidence to look into that.
Okay, thank you. No more questions, but I have a question. Can you shed some light on molybdenum and sulfate antagonism?
Yeah, so this was like mainly, so antagonism, it's happened when there are more sulfate, because as you have seen by third slide probably, yeah. So there was like a molybdenum as one of the cofactors for this enzyme, so this, because they do the MOFE clusters. So when there are more sulfate that could bind instead of molybdenum, like that is also happened mainly in the upwelling zone. And in this context also, like when there is molybdenum is not able to bind, even in the lower temperature, mainly in the upwelling, there are prevalence of other nitrogenous enzyme. So these are called the alternative nitrogenous that act as a backup enzyme system. So when there is the main conventional nitrogenase is forced to work, like this MOFE nitrogenase, there are two other nitrogenase, like it's called vanadium and iron only nitrogenase that could bind in this part, and it could back up and act as like a main enzyme system. That also happened like in the, if you look into the Benguela and Peruvian upwelling system, there people found the traces of the alternative nitrogenase.
Following this question, I would like to ask further.
Yeah.
In your future cruises or maybe missions, will you be trying to assess the effect of such molybdenum sulfate antagonism on limiting nitrogen fixation in any region?
Yeah, I tried even in the southwestern part, and there is an influence of the, but I have only the particular data, don't the dissolved data. So even the particular data that molybdenum is showing a strong effect. And there is also this factor that Taneja was asking. So molybdenum in particular, I have the like a particulate form of the data, and they suggested that this molybdenum has a strong influence. And this could also explain in the future about the northern part of the Indian Ocean, because there was a paper that highlighted that there are more molybdenum in the Arabian seaside than the Bay of Bengal. So that can also explain in future if we even look into the consideration like why there are more nitrogen fixation activity in the Arabian Sea, not in the Bay of Bengal.
Okay. So with this, we conclude this talk. There are no more questions. Thank you so much, Subhadeep, for accepting our invitation, for presenting your recently published paper. Thanks a lot. And I hope you all will join in next seminar in November, first week Friday. Thank you.
Thank you everyone for your attention. Bye-bye.